Hello and welcome to this Revision Monkey video on what's going to be in the 2022 exams. This video is for AQA Combined Scientists. So do double check with your teacher that you're watching the right video. There's two types of combined science. The majority of people will do the Trilogy one, which is the focus of this video. And this video is for Biology Paper 1 and it's for higher tier students. So first of all, let's go through what's not going to be assessed on the paper. And these topics are microscopy, transport in cells, plant tissues, organs and organ systems, viral diseases, fungal diseases, protist diseases, human defence systems, uses of glucose from photosynthesis, and response to exercise. Next, the exam board told us what required practicals will be in the exam this year, and those are food tests, enzymes, and photosynthesis. So what I'll do for you is I'll do a video just on these required practicals, and I'll add the link into the description for this video. So do look out for that. I'll edit you a video which is just focusing on these required practicals because this is what we've been told will come up this year. We've also been told what topics will be the major focus of the exam. So those are cell division, animal tissues, organs and organ systems, and photosynthesis. So these are the topics that I'll focus on for this video. And also keep an eye out in the description, and what I hope to do is add a link to some questions for you based on these major foci the examiners have told us about. So in this topic of cell division, we need to know a little bit about chromosomes. So plant cells and animal cells both have a nucleus. And if we zoom into a nucleus here, the nucleus of a cell contains chromosomes. And the chromosomes themselves, if they're unravelled, will show this really long DNA molecule. So the chromosomes are within the nucleus, and the chromosomes are made up of a DNA molecule. Each chromosome carries a large number of genes, and a gene is a small section of a chromosome. So we could, for example, measure this length of DNA here, and this could code for a particular gene. In body cells, these chromosomes are found in pairs, so it will be different for different organisms, but for example, humans have 23 pairs of chromosomes in each body cell. So this topic is delivered in a lot more detail in topic B6, which comes up in paper 2, but for paper 1 they just briefly touch upon the fact that the nucleus contains chromosomes, 23 pairs of those in humans, and that's made up of DNA molecule, and that a particular sequence of DNA is called a gene. So the type of cell division that we're going to be talking about in paper 1 is mitosis. So this one must be spelt correctly because it sounds very similar to meiosis, which comes up in paper 2. So mitosis is the cell division we need to focus on, and that's for growth and development in multicellular organisms. And essentially we're talking about one body cell replicating to form two identical cells. And for this process, the cell goes through a process called the cell cycle, which is depicted here. It has 3 stages. The first is growth and DNA replication. Next, it's mitosis. And then finally, the cell divides. So in the first part of this cell cycle process, because we need to produce 2 cells, we need to increase the number of subcellular structures, for example, the ribosomes and the mitochondria. The subcellular structures just mean the things that are inside the cell. So here we've got the ribosomes, the mitochondria, the cytoplasm, everything like that needs to double up essentially because we need to produce 2 cells from 1. So this takes the longest amount of time, to increase all these subcellular structures. Also, as part of this process, we have DNA replication, whereby the DNA is replicated to form 2 copies of each chromosome. So in the nucleus of the human body cell, there'll be 23 pairs of chromosomes. So pair 1, pair 2, all the way down to 23. And then these chromosomes double up, so they look a little bit like this. So each arm is identical, there's the same genes in each one, we're just replicating the DNA. And then that will be ready for mitosis. So every single pair from pair 1 all the way down to pair 23, the DNA would be replicated to form 2 copies of each chromosome. So after the cells have gone through the growth and DNA replication stage, they've got more ribosomes and mitochondria and cytoplasm, so they're a bit bigger. So in human body cells, we'd have 23 pairs of chromosomes, or 46 in total, and they have now got an identical chromosome attached to them. So they would all line up in the middle of the cell, like so, all the way down from number 1 to number 46, if we're talking about a human body cell. And then this next stage is mitosis, and in mitosis, one set of chromosomes is pulled to each end of the cell and the nucleus divides. So here we get one arm of the chromosome, exactly identical, pulling to one edge of the cell and the other one going in the opposite direction. So these chromosomes get pulled apart, but you can have an identical arm there, as they get pulled apart like so. So then over here we'd have one nucleus forming with 23 pairs of chromosomes in them, and we have another nucleus forming over here with 23 pairs of chromosomes, so 46 altogether. So in here we'd have 46 chromosomes, in here we'd have 46 chromosomes. If we were talking about a human body cell, if we're talking about another organism, then this number would be different. So that's the smaller part of the cell cycle here. After they've gone through the growth and DNA replication, mitosis is when these chromosomes are getting pulled apart and the nucleus divides. And then the final part of cell division is the cells actually dividing. So this final part here. So after mitosis, the cytoplasm, which is the jelly-like stuff in the middle of the cell, and the cell membranes on the outside will divide. So that will end up splitting up into 2 identical cells. The last part of the cell division topic is on stem cells. Stem cells are undifferentiated cells, which means cells that can turn into many different types of cells. For example, when a human organism is first created, the sperm meets the egg, and you end up with one fertilised cell. And this cell divides by mitosis to form a small ball of cells, which we call an embryo. And at this point, these embryonic cells are stem cells. They are undifferentiated, which means they can turn into any type of cell, which of course, if we think about it, this ball of cells here is ending up going to create a full human organism with all the muscle tissues and bone tissues and blood, etc. So at this point here, these cells can turn into any type of cell. That's what undifferentiated means. And at the embryo stage, we call these embryonic stem cells. Okay, so in humans we can find those in embryos. But also in your adult life, you have some stem cells in your body in the bone marrow. These aren't as good as the embryonic stem cells because they can't turn into any type of cell, but they can produce things such as red blood cells for us. So the embryonic stem cells are the best ones. They can turn into any type of cell. The bone marrow that we have as adults has some limited uses. Plants also have stem cells. In plants, this is found in the meristem tissue. And there's lots of different places in plants that you find the meristem tissue. You find them in the tips of the shoots and also in the roots of the plants. And you'll also find them at various points in the stem as well. So this plant tissue is undifferentiated, so they can turn into any type of cell. And with plants, this undifferentiated cells remain throughout the whole lifetime of the plant. So as the plant grows and develops, you've got stem cells in there which can then produce the bark, for example, the flowers, whatever type of tissues you need on your particular plant. Whereas in adults, we have these ball of undifferentiated cells when we are at the embryo stage, but as we develop into human organism, our stem cells are much more limited. We only have them in our bone marrow to make a few different types of cells, whereas plants remain, the meristem tissue remains undifferentiated for the whole lifetime of the plant. Stem cells are really interesting for scientists because they are undifferentiated. Scientists are trying to use them to produce lots of different types of cells that can treat certain disorders. For example, they're looking into them for paralysis. So they're thinking at this stage, when we have an embryo, could we take a cell out, get it to produce nerve cells, and use that to treat someone who is paralysed? Same with conditions such as diabetes. When a patient can't produce insulin, for example, they're thinking, well, at this embryo stage, could we turn this on to produce a cell which produces insulin for the patient and transfer that into the patient? So these are the kind of the ideas that are surrounding stem cells at the moment. And for scientists who work with plants, the fact that there's meristem tissue that remains undifferentiated throughout the whole lifetime of the plant means that if you find a really rare plant, you can clone that plant to protect it from extinction. So because there's meristem tissue in the plant, you can take cuttings of the plant, for example, cut that one there, put it in some nutrients, and that will grow into a new plant for you. So you can clone them really quickly if you've got only a few examples of rare plants left. And also you can clone desirable crop plants really quickly. So for example, if a farmer had a plant that was particularly resistant to disease or drought, or perhaps they produced a genetically modified plant, they can clone them really quickly because of this meristem tissue that's throughout the plant. So it's undifferentiated, so you can take cuttings, grow them in nutrients and clone them really quickly. Where the stem cell technology is going ahead with humans is in something called therapeutic cloning, so using cloning for treating disease. And with therapeutic cloning, an embryo is produced, so this ball of cells down here, with the same genetic information as the patient. And how they do this is they take the body cell from a patient and they take out the nucleus, because that contains the DNA. They use an empty egg cell from a donor. So a woman would have donated an egg cell, scientists would have removed the nucleus, so you just have the empty cell. You put the nucleus of the patient into the cell and you'll stimulate that to divide to make an embryo. So this is really interesting because obviously if we let this embryo continue to divide, if we were to implant this inside a woman, it could develop a clone of this human here whose DNA we have removed. So it's really important that that doesn't happen. Otherwise we'll be cloning humans and that's completely unethical. But the idea of this is that can we use this embryo containing the patient's DNA to produce some cells for the patient that won't be rejected? So perhaps they need some nerve cells or some muscle cells, or perhaps they need some bone cells or cells that produce insulin. And if they use stem cells with their DNA, then that's less likely to be rejected from the body. So you could well be asked an evaluate question on therapeutic cloning. For example, evaluate whether a patient should be treated for nerve damage using therapeutic cloning. In any evaluate question, you should give advantages and disadvantages and finish with what we call a justified conclusion. So I'll just go through this example with you. If we were evaluating whether we should use therapeutic cloning for nerve damage, our advantages could be things such as stem cells contain the patient's DNA, so will not be rejected. The patient will have a better quality of life. And perhaps that there'll be no long-term NHS costs to treat nerve damage, because if we fix the person's disorder, then they wouldn't have to have drugs and treatment on the NHS. Disadvantages could include the stem cells could mutate and cause illness. So as the stem cells are dividing, they could mutate, and when we put them into the human that needs treating, that could cause them an illness. The stem cells, because they're dividing lots, they could actually be contaminated with a virus. Okay, so a virus could attack the cells whilst a scientist is working on them, and then this could be transferred to the patient. And ethical ideas, for example, it isn't ethical to be using and destroying embryos. So there's a lot of debate around at what point you should consider this ball of cells as being a human life, because we'll use the cells and then destroy the rest of the embryo. So once you've given advantages and disadvantages, you must use a justified conclusion. For example, you could write, overall, I believe therapeutic cloning should be used because. So state whether you are for or against it. There's no right or wrong here, just choose one particular stance. And then try and use the word although. Although there is a risk the patient could catch a virus and there's no guarantee it will work, without the treatment, the patient will suffer a lot throughout their life, so it's worth a try. So this is just an example of a justified conclusion. You give your stance whether you are for or against it, so I've said it should be used, and then by justifying you're saying, although some of these are the bad things, overall it will be better because. This topic is about animal tissues, organs and organ systems. This image is of the digestive system, and you're expected to know the organs that are in this system and what they do. So when we eat, we take in food in our mouth. The mouth also has some glands in it called salivary glands, which are involved in the digestion process because they produce enzymes, which we'll talk about later. The food goes down your gullet, otherwise known as your esophagus. Esophagus is the word they're likely to use in the exam. And then it goes into your stomach, which is over here. Your stomach contains an acid called hydrochloric acid, which I'll write as HCl. That kills any bacteria that you consume when you eat the food. And the stomach also churns up the food and aids in the digestion process. After the stomach, the food goes into your small intestine, which I'm just going to abbreviate to SI here. Your small intestine, that is where most of the digestion happens. It has folds called villi, which are providing a large surface area for digestion of small soluble molecules, molecules that can dissolve and move across the membrane. So these folds in the small intestine, these villi have a close network of capillaries next to them as well. So there'll be capillaries nearby and wrapping around so that small molecules can move out of the villi into the bloodstream, things like glucose and amino acids. After the small intestine, your food will go into the large intestine, which I'm just going to abbreviate to LI, and this is where excess, which is the extra water, is absorbed. So it's a bit of a sloshy mess when it goes into your large intestine, and then any excess water is absorbed back into the bloodstream, and then it will start to form the feces as it goes around. That feces will get stored in the rectum and finally will be excreted from the anus. So that's the food, that's the organs that the food moves through. You also get some other organs involved in the digestion process. This yellow organ here, hiding behind, is called the pancreas. And food does not go through the pancreas, but it is involved in digestion because it produces the enzymes, which we'll talk about later. Secondly, the liver, which is this large organ here, and the gallbladder, which is just next to it, are both involved in digestion. However, the food does not go through these organs. These two organs are involved in digestion because of bile. The liver produces bile and it is stored in the gallbladder. And bile is a chemical that does two major things. It neutralises stomach acid, and this has a link to enzymes because when the food goes into the stomach, it goes into this acid and is covered in acid. As it moves into the small intestine, you do not want that acid going with it because the enzymes in the small intestine really like an alkaline environment. So that bile neutralises the stomach acid, which helps with that enzyme activity in the small intestine. And also it emulsifies fats. And what that means is it will break a large lump of fat up into smaller droplets like this. And in doing so, it increases the surface area, which I'm just going to abbreviate SA, increases the surface area of that fat so that it is more easily digested when it reaches the small intestine. Within the digestive system, enzymes play an active role in digestion. This is an example of what one might look like, just a model of one. You've got a couple of important parts. This is the enzyme, and the face of the enzyme here is called the active site. And each enzyme has a different shaped active site to fit a particular molecule, and we call the molecule that's fitting in there the substrate. So for example, this could be a long starch molecule that's coming in to an enzyme which will break it up into smaller pieces. So the enzymes are example of biological catalysts. They are catalysts because they speed up chemical reactions, and it will be different enzymes for different reactions because of their shape of their active site. So you can see that here the substrate is a particular shape to fit in the active site. So we describe this as being like a lock and a key, whereby the enzyme and the active site of the enzyme in particular is the lock, and the key that fits into that is the substrate. So it's called the lock and key theory because the enzyme has a particular shaped active site that fits a particular shaped substrate. So this is the most simple model for representing an enzyme, the lock and key model. There are also other models. For example, there's one called Induced Fit. And with this, the active site is a particular shape again to fit the substrate, but as the substrate comes into the enzyme changes shape slightly just to fit and grab that substrate a little bit better. So the main one to remember is lock and key, but another one is induced fit. When the substrate goes in, the active site changes slightly to grip that molecule in there. So we said enzymes are biological catalysts, so they affect the rate of reactions, they help speed up the rate of reactions, and they are molecules that are subject to working best at particular temperatures and pHs. Temperature inside the body is maintained at 37 degrees C. So what you'll see with enzyme activity is you'll see a graph that shows that as temperature increases, the rate of a reaction increases. There'll be an optimal temperature and then it will drop off. So optimum means best. And inside the body, the enzymes will work best at around 37 degrees C, because that's the temperature inside the body. The same with pH, but this might be different depending on whether the enzymes are working in the stomach, which is acidic, or whether it's working in the small intestine, which is alkaline conditions. So again, there'll be an optimum pH for enzyme activity, so a rate of reaction will increase and then decrease, and there will be an optimum pH for each particular enzyme to be working at. The reason why optimum temperature and pH are important for enzyme activity is because if we go beyond their optimum, for example here if we have the enzymes at high temperatures, then that causes this active site to change shape. And if this active site changes shape, it will no longer bind the substrate. So here at high temperatures, the enzyme denatures. Okay, we don't say that it is killed or it dies because it's not living, it's just a protein, it's not a living thing. So enzyme denatures, we have to say instead, and what that means is the active site changes shape and that means the substrate will no longer bind, because it has to be like a lock and key, a very specific shape, and if the temperature gets too high, the enzyme denatures and the active site changes shape. All enzymes end in A's, so you can remember that if it's got an A's on the end, that's an enzyme. The three main ones that we're going to discuss are amylase, protease and lipase. Proteases and lipases are groups of enzymes, and amylase is a particular enzyme which actually belongs to the group carbohydrases. Carbohydrase is any enzyme that breaks down carbohydrates into sugars. And the particular one that you'll see talked about most in the exam is amylase, which breaks down particularly starch into sugar. So you may well have learned about carbohydrases. They break down any carbohydrates into sugars, and amylase is just a particular carbohydrate. Amylase is produced in the salivary glands and in the small intestines, and it also works there in both places. Protease is produced in the stomach and the small intestine, and it also works in those two places. And lipase is produced and works in the small intestine. So you'll see most of the digestion is happening in the small intestine because all three enzymes are working in there, breaking down the large molecules into smaller ones. Amylase breaks down starch into sugar, protease breaks down protein into amino acids, and lipase breaks down lipids, which are fats, into fatty acids and glycerol. All three enzymes are also produced in the pancreas. Digestion doesn't happen in the pancreas, but the pancreas will produce these enzymes and then transfer them into different organs of the digestive system. So within this small intestine here, the folds of the small intestine have a large surface area. They have lots of folds called villi. Anytime in science you see a shape like this, I want you to always think of the fact that that has a large surface area, which I'm just going to shorten to SA for short, large surface area. And this is accompanied by a close network of capillaries, so that the food molecules that have been broken down into small pieces can pass from the small intestine into the bloodstream. So for example, your sugars, your amino acids, your fatty acids and your glycerol are really small molecules that can be transported into the bloodstream and then passed around the body. And then they can be used as part of your metabolism, which is the sum of all the chemical reactions in your body. As part of your metabolism, one of the things that will happen is these small molecules, when they're transported to different areas of the body, they might be turned into large molecules again. For example, if we had the fatty acids and glycerol combining again, you have one glycerol with 3 fatty acids, and that would make a lipid molecule, and that can be used to deposit a layer of fat in your body, for example. And some of the sugar will be used straight away for respiration, which happens in all cells of your body. One of the organs they'd like you to focus on is the heart. The bottom chambers of the heart are called ventricles, and the top chambers of the heart are called atria. So as you look at images of the heart, you have to imagine it as if you're looking at it inside a person. So this side of the heart is actually the left, and this side of the heart is the right. So we have a left atrium here and a right atrium here, but the plural is atria. So inside the right atrium, you have some special cells called pacemaker cells. And the pacemaker cells are a group of cells that control your natural resting heart rate. So anybody that has a disorder affecting their natural heart rate, if they've got irregularities with their resting heart rate, they may well have an artificial pacemaker installed. And what that is that's an electrical device which will correct the heart rate, correct any irregularities in your heart rate to bring your natural heart rate back to a regular beat. There are 4 major blood vessels that we need to be aware of. The first one is the aorta. The aorta takes blood from the left ventricle here out away from the heart to the rest of the body. So because it's moving blood away from the heart, that is an artery. Artery moves blood away from the heart. And this left ventricle actually has a very thick wall, much thicker than the right ventricle, because this has to push the blood all the way around the body. And when the blood goes around the body, oxygen will diffuse out of the blood into other cells so that they can use it for respiration, and the blood will return back to the heart as deoxygenated blood through the vena cava. So the vena cava is the main vein that brings blood back to the heart. So arteries bring blood away from the heart, veins bring blood back to the heart. So blood will come in to the vena cava like so, and into the right ventricle. But now this blood doesn't have very much oxygen in it because it's given it to the other cells in the body. So the next place the blood needs to go is back up to the lungs, and it does that out of the pulmonary artery. So the blood goes out of the pulmonary artery into the lungs. So anything to do with lungs is called pulmonary, and it's an artery because it's taking blood away from the heart. So it goes out of the pulmonary artery to the lungs, where gas exchange will take place and the oxygen will be diffusing back into the blood. And then finally, from the lungs, the blood will go through the pulmonary vein back into the heart. So pulmonary because it's to do with the lungs, and vein because it's coming back to the heart. And then that will go into the left ventricle and out of the aorta again. So you'll see we've talked about two circuits. If this box is the heart, just put a H for the heart, we've talked about one circuit via the aorta taking blood to the body and the blood returning through the vena cava back to the heart in one circuit. And we've talked about blood leaving through the pulmonary artery to the lungs through the pulmonary artery and then back from the lungs through the pulmonary vein to the heart. So you can see in humans we've got two circuits. We've got one circuit going to the body, one circuit going to the lungs, which is why it is called a double circulatory system. Inside the heart, between the atria and the ventricles, you have valves which control where the blood is positioned within the heart at different times. So they open and close, allowing movement from the atria to the ventricles. And also, finally, not shown on this diagram, but the heart itself needs a supply of oxygen. So the heart itself is covered in coronary arteries, which supply the heart muscle with its blood supply. So these are coronary arteries, and these are the ones that can be blocked up with cholesterol deposits and cause coronary heart disease. So the three main parts of the lungs we need to know about are the trachea, which is the windpipe going into the lungs. This branches out into two branches called the bronchi. So that's the plural, the bronchi. So this one here is pointing to the left bronchus, because again, like the heart, we look at it as if it's inside the person. And over here we've got the right-hand side, so this would be the right bronchus here. And then it goes down small tubes called bronchioles, which you might have learned about in Key Stage 3. But the other key thing we need to know about is the alveoli, okay? So the alveoli is plural. You might see the word alveolus, which is one singular alveoli. So the alveoli are the air sacs at the end of the lungs where gas exchange takes place. Again, key thing is the fact that they're shaped to provide a large surface area, which I'll just abbreviate to SA, for gas exchange. You need to know that there is a close network of capillaries around the alveoli so that gas exchange can take place. So they'll have some red blood cells in there, and oxygen will diffuse from the alveoli into the blood. And carbon dioxide, which is in the blood plasma, will diffuse in the other direction out of the blood into the alveoli. And that's why they call it gas exchange, because oxygen is being passed into the blood, and in exchange for that, carbon dioxide is being passed out of the blood by the process of diffusion. So the alveoli have a large surface area. They have thin walls to allow this diffusion to happen very quickly. They also are moist, and that just helps with gas exchange. If there's a big air gap between the alveoli and the carbon dioxide, that'd be very difficult for the gases to pass across. So having them moist makes that a lot easier. The three blood vessels that we need to be aware of are the artery, the vein, and the capillary. Artery, as we said, takes blood away from the heart, veins take blood to the heart, and capillaries are very small blood vessels that are used in places like the muscles and the lungs to transfer things such as gases and glucose and things like that to where the body needs it. So the arteries have thick muscular walls and elastic fibres within those walls, and this is because the blood flows at very high pressure through the arteries. It's getting squeezed by the ventricles away from the heart at really high pressure, so it's got to have these thick walls so that they don't burst open. The veins are taking blood back to the heart, so they have thinner walls. They have valves, which is shown here, to stop the blood flowing back down the veins. And in the veins, blood flows at lower pressure, which is why they can have thinner walls, because it's less likely to be bursting out. And they have a large lumen. The lumen is the bit inside. So this is larger on the vein and smaller on the artery. The capillaries are really tiny. They have very, very thin walls, and they are just one cell thick. And this is where, for example, when you get to a muscle, having thin walls will help with gas exchange of oxygen into the muscle and carbon dioxide out. So capillaries are really, really, really tiny. The liquid in the blood is called plasma. And plasma carries a number of different cells, for example, the red blood cells, the white blood cells, and small fragments of cells called platelets. The plasma also carries dissolved substances, things like carbon dioxide that is dissolved in the plasma, perhaps things like glucose, and also hormones that travel in the blood. The red blood cells have a pigment in them called hemoglobin, and that hemoglobin binds oxygen. So when gas exchange takes place in the lungs, the oxygen binds to the hemoglobin and the red blood cells transport that around the body. The white blood cells are part of our immune system, and they help to defend us against disease. And the platelets are fragments of cells which help to clot the blood to prevent you from losing lots of blood if you cut yourself. The heart itself is covered with coronary arteries, which provides the heart with oxygen. The coronary arteries need to be open to allow the blood to flow properly around the heart. However, if people have a poor diet and a lack of exercise, for example, a high fat or a high cholesterol diet, that can cause fatty deposits to build up inside the arteries. And if those fatty deposits build up, what that means is there's a reduced volume inside there for the blood to flow. And then it puts a lot of pressure on the walls of the artery, and it can cause a lack of oxygen to the heart, and this can ultimately result in a heart attack. So there's two ways you can resolve these blocked arteries. One way is using a stent, which is a wire mesh that's put inside the artery, which will expand and open up that lumen, so you've got an increased volume for blood flow. Or you can use statins, which are drugs which will help reduce the cholesterol and therefore reduce these fatty deposits. So you could be asked an evaluate question in the exam where you're looking at the pros and cons of stents versus statins. So let's go through some of the main advantages and disadvantages of stents. Advantages include the fact that they lower the risk of a heart attack. Surgery is quick and the effect is instant. Disadvantages include the fact that there is a risk of heart attack during surgery and that there's a risk of infection or blood clots after surgery. With the statin approach, the advantages are that it increases good cholesterol and lowers bad cholesterol within the body. It can prevent other diseases as well. So by taking these pills, it's turned out that actually they can be good at reducing other diseases as well as the coronary heart disease. Disadvantages include that you have to remember to take them regularly. So like any other medicine, you have to remind yourself to take them, and if you forget, then that could cause the problem to get worse. And the effect of statins is not instant. So with the stent, once you've had the operation, the result of that is instant, but statins, it might take a lot longer. Another problem that people can have with their heart is the valves in the heart. They control the blood flow from the atria into the ventricles and then the ventricles out again. So it's important that the heart is not flooded with blood. It's important that it's kept in its correct chamber at each part of the heartbeat. And if these valves become faulty or leaky, then you can have a real problem with the function of your heart. It's trying to work harder than it should do to get that blood around your body, and it can ultimately result in a heart attack. So you could need your valves replacing. You could have a biological replacement, which could be a human valve replacement, or you could have a valve replaced by a cow or a pig, or you could have a mechanical valve put in instead. So rather than being made out of something biological, it will be a synthetic material to replace that valve. However, with both of these, you can have risk of infection, a risk of heart attack during surgery. You could also have risk that these valves are rejected by your body. Some people going into heart failure might well need to wait on a heart transplant list, and this might involve a full transplant of the heart. And again, obviously this has lots of risks with the heart potentially being rejected by your body and risk of infection and a very difficult, complicated surgery as well. But whilst people are waiting for surgery, sometimes they are asked to use an artificial heart. So you can see this gentleman here, he has got tubes running into a backpack here, and in this backpack there is an artificial heart, which is a pump helping his blood flow properly around the body, because essentially all the heart is a pump. So whilst he's waiting for the transplant, he's using this artificial heart, which obviously has problems as well because it's a mechanical and electrical device, so that has risk of failure as well. The last part of this unit looks at health and disease. Health is the state of physical and mental well-being, and the type of disease discussed in this topic is non-communicable disease, which is disease that cannot be spread between organisms. So they're disorders that you can't catch from each other. So things like diabetes and cancer you can't pass on to other people. But there are risk factors that could increase your likelihood of getting a non-communicable disease. Risk factors could be lifestyle or environmental risk factors. And risk factor is anything that increases the likelihood of a disease. So lifestyle things could be things such as smoking, obesity, alcohol, and your diet, high fat or high salt diet. And risk factors for the environment could be things such as air pollution where you live or where you work, how much UV exposure you're getting, for example, if you had a job where you're always working outside, and perhaps something specific, for example, if you have a job working with asbestos, which is a chemical that can cause a disease called asbestosis. So having these risk factors can increase your chances of certain disorders. So it's important for the exam that you're just made aware of a few of these. So I'll run through a few now. So for example, if you're exposed to air pollution, you could get some respiratory problems, High UV exposure could lead to skin cancer, and asbestos could link to asbestosis. So let's now look at some of those lifestyle risk factors. So for example, smoking and alcohol can lead to disorders in unborn babies. If you smoke or drink alcohol during pregnancy, you can get underweight babies, you can get babies with fetal alcohol syndrome, for example. Alcohol consumption can also lead to liver and brain function, so liver disease, for example, cirrhosis of the liver, problems with your brain. Obesity can lead to type 2 diabetes, can also put lots of pressure on the heart and contribute to heart disorders as well. Smoking can cause cancer and lung disease. Smoking, obesity and a high-fat diet are also contributors to cardiovascular disease. So there's lots of these risk factors that increase your likelihood of getting certain diseases. As well as these lifestyle and environmental risk factors, it's becoming increasingly the case that your genes can actually increase your likelihood of getting certain disorders. For example, they're looking at genes that increase your chances of getting breast cancer, for example, and some women are screening to find out if they have that gene and are perhaps opting for preventative measures to stop them getting the breast cancer, like removing their breast tissue to reduce their risk of actually getting breast cancer. So as well as your lifestyle choices, your risk factors, environmental risk factors, actually your genes can play a part in increasing the likelihood of you getting certain disorders. Diseases can also interact with each other. That means if you get one disease, that can also increase your likelihood of getting another disease. For example, if you already have a weakened immune system, you're more likely then to suffer other infectious diseases. If you have viruses in your cells, that can be a trigger for some cancers. For example, the HPV virus in women can be a trigger for cervical cancer. Immune reactions caused by a pathogen can also trigger allergies or rashes or asthma. So if your body is responding to a particular bacteria or virus or other pathogen entering your body, that can cause other issues as well. And also severe physical health can lead to mental health issues. For example, if you've broken your leg, you're out of work for a long time, you can't see your friends, you're in the house a lot of the time, you're not very mobile, this can then lead to depression, anxiety and other mental health issues as well. Cancer can be defined as changes in cells that lead to uncontrolled growth and division. And like we said before, there are some genetic risk factors for some cancers. For example, scientists have identified a gene that increases your likelihood of getting breast cancer. So some people will have genes that make them have a higher chance of getting certain cancers. You can also get cancers because of your risk factors, so smoking, obesity, UV exposure, for example, can increase your likelihood of getting cancer. Some viral infections, like we previously mentioned, like HPV, is linked to cervical cancer. If you get hepatitis B or C, that can increase your chance of getting liver cancer, for example. So there's lots of risk factors associated with cancer. Hence, at the moment, we're talking about 1 in 2 people in your lifetime ending up having some form of cancer. So cells divide naturally by the process of mitosis for growth and repair. So in the body, they'll be replenishing and replacing their cells by mitosis. But cancer is when that cell division starts to get out of control and the cells are dividing uncontrollably. And this can lead to a tumour. Now, there are actually 2 forms of tumours. There are benign tumours and malignant tumours. With benign tumours, the cell division is often encased within a bigger membrane, so the tumour actually stays in one place. So this could just be a small lump on somebody, but stays in one place and doesn't move anywhere else. These are often tumours that can be removed without any problem to the patient. Malignant tumours, however, in malignant tumours, the cells that are dividing uncontrollably can actually break away and enter into the bloodstream and travel to another place in the body where they will make a secondary tumour. Okay, so malignant tumours, the cells can break off, travel through the bloodstream and invade other healthy tissues. And this type of tumour is our cancerous tumour. So benign ones, most of the time you can just take them away without any issues, small surgery, whereas malignant tumours, because those cancerous cells can spread around the body, that's when it is more difficult to treat. The last part of the video shows the information you need for the last major focus, which is photosynthesis. This is the word equation for photosynthesis. Carbon dioxide plus water makes glucose and oxygen. As a symbol, it would be CO2, add H2O, makes C6H12O6 plus O2, which we'd balance by putting a 6 there, a 6 there, and a 6 there. So you need to know both versions. This takes place in plants inside these structures here called chloroplasts. Within the chloroplast there is a green pigment that is called chlorophyll. And it is the chlorophyll that absorbs the sunlight needed for photosynthesis. So sometimes on top of these arrows you might see sunlight and chlorophyll to show that both of these things are needed for this process. But ultimately the goal of photosynthesis is to produce glucose that the plant then uses as its food. This reaction is an endothermic reaction linking to the chemistry topic because it takes in energy. There are several factors that can affect the rate of photosynthesis that we call limiting factors. Those three limiting factors are light, carbon dioxide levels, and temperature. Those are the main ones, but also the green pigment, chlorophyll, is also a limiting factor that can affect how much photosynthesis takes place in the plant. Chlorophyll becomes a limiting factor when the plant has a disease, because disease like tobacco mosaic virus or rose black spot fungus, that disease affects the amount of chlorophyll in the plant, and chlorophyll absorbs the sunlight for photosynthesis to take place. So if a plant is diseased, then chlorophyll will come in as a limiting factor. But the main ones are light, carbon dioxide and temperature. So light and carbon dioxide have a very similar shape graph, whereby as light increases, the rate of photosynthesis increases up until a particular point, and then the rate will not increase further and it will level out. The same with carbon dioxide. As the rate of photosynthesis, the amount of carbon dioxide increases, so does the rate of photosynthesis until it levels out. This initial point of the graph here, while it's still increasing, this is when, in this case, light is a limiting factor. Or in this case, carbon dioxide, this is the point where it is a limiting factor. That means if you were to increase or decrease the light levels at this point, you would change the rate of photosynthesis. But after that, it doesn't matter. If you were to invest more in more lighting, well, that wouldn't increase the rate any further. So at this point, we would say that light is not a limiting factor because it won't change the amount of photosynthesis that takes place. So this is not a limiting factor at this point here, and the same for carbon dioxide here. Temperature has a different shape curve, and that's to do with enzymes that control the rate of photosynthesis. So this here would show the optimum or the best temperature for photosynthesis to take place at, And then after this, the enzymes denature and the active site changes shape and they will no longer work. So up until this point here, temperature is a limiting factor, and then after the optimum temperature, the enzymes denature and no longer work. So this would be important in the winter. Temperature is the obvious limiting factor in the winter, and at night time, light is a limiting factor because without the light you don't have photosynthesis. This extra slide is for higher tier only because you need to know how these factors can interact with each other. For example, if we had a graph that showed light intensity against rate of photosynthesis, before we just had one of these lines on the graph. So before we just had this one, for example. But you may well see in the exam graphs with two different lines on, and this could have a different key to show you that this one perhaps was at 15 degrees, for example, and this one was at 25 degrees C. And what this will show is that in this case, temperature was the limiting factor, because if you increased the temperature to 25, you increased the rate of photosynthesis. So temperature was a limiting factor affecting the rate of photosynthesis. Here we had one at 15 degrees that plateaued out, but if we increase the temperature, then the rate of photosynthesis also increased. This is not a difficult concept, but for higher tier only, you also need to know a little bit of detail about how greenhouses are managed. So greenhouses are glass buildings that are used to grow lots of plants, and the main trade-off that you have when running a greenhouse is the cost of running the greenhouse versus the crop yield that you will get inside the greenhouse, whether that is the flowers that you're growing or the vegetables you're growing or whatever it is. So you want to maximise the rate of photosynthesis that's taking place within the plants inside the greenhouse, but at the same time not be wasting your money when you're doing so. So this will link into limiting factors and the things that will affect the rate of photosynthesis. So obviously the outside is glass to try and let lots of light in, but you also might want to put artificial lights inside the greenhouse to increase the rate of photosynthesis. Obviously they're going to come at a cost as well. You can also help to heat your greenhouse, especially important in the winter, if you were to put a special heater inside called a paraffin heater. And this does two things. It will help maintain the optimum temperature for you, which is important, but the paraffin heaters also release CO2, so they'll help optimise the CO2 levels inside the greenhouse for photosynthesis. You can also have windows like these at the top, this top layer here, that can automatically open and close to adjust the temperature to make sure that is optimal. And of course the fact that it is all in one enclosed building will also reduce the number of pests inside, and obviously that is a added benefit for you when you're trying to grow plants inside. So think of perhaps an evaluate question associated with this, looking at the trade-off between the cost of these things and also the effect that they will have on photosynthesis and crop yield. Thank you very much for watching. Please do look out for the required practical videos for 2022 and practice questions. I'll add these links to the description as soon as they're done.